So aloha everyone and welcome. My name is Lisa Canali and I'm the coordinator of the University of Hawaii at Hilo Tropical Conservation Biology and Environmental Science Graduate Programs Professional Internship Track. Quite a mouthful. One of the requirements of the graduate program is a public presentation, and today we have the great fortune to learn about the work of Keena Curbelo. Keena Michelle Curbelo was raised on the north shore of Oahu before going on to receive her BS in biology from Skidmore College in Saratoga Springs, New York. She then returned to Hilo, to Hawaii, to pursue her master's here in our program. She works currently as a biological science technician for the U.S. Department of Ag, studying insect pests that affect farmers across the country. Her free time is spent with her husband, her two dogs, who we get to see in the background there, and her horse, who is not in the room. But on a more personal note, I first met Keena via email in May of 2019, and I've had the good fortune to have her both as a student and to be her advisor. And I must say, Keena is an outstanding student. She constantly, consistently presents great work. She's on point when she contributes to the class. And as a professional, she has drive and purpose. And I'm really happy that you're all here to spend the afternoon to support her and learn about her work with the Queensland's Longhorn Beetle. And so now I'd like to hand over the Zoom mic to her mentor.
Hi, Lisa. Everyone. Thank you so much for organizing this and for everyone who's in attendance. And thank you to Keena for putting in all the work to make this presentation happen. So Keena joined my program in 2018 when I first started as a new research biologist with the USDA ARS. And since then, she and I have helped each other grow and learn and build a research program together. And I'm really glad that we can work together with mutual respect and fondness. I really appreciate all of the additional responsibilities she's taken in the lab, particularly with this new beetle project. And I really appreciate all the work that she's contributed to the basic understanding of this new to us invasive species. So with that, I'll turn it over to Keena, who will now be presenting her work on characterizing the Acalolepta aesthetica invasion in Hawaii.
Thanks, Shanna. Thanks, Lisa. Really appreciate that. So hi, everyone. My name is Keena Curbelo, and today I will be presenting some of my work on the Queensland longhorned beetle. This work was done at the United States Department of Agriculture Agricultural Research Service Pacific Basin Agricultural Research Center in Hilo, Hawaii. I know it's a little bit of a mouthful. A note on the photos in this presentation. Photos taken by me will not have a photo credit included. However, I have included the names of the photographers on any other photos. Also, you will notice some really fun beetle animations, which my talented supervisor, Shenna Sim, created herself in order to better illustrate the life cycle of this pest. Dr. Shenna Sim is a research scientist in the Tropical Crop and Commodity Protection Research Unit here in Hilo, and has served as my mentor on this project. So, for a little background, I am a biological science technician at the USDA Agricultural Research Service. The ARS is the research arm of the USDA and is composed of almost 100 different locations. This is a photo of our research center. It's located just up the hill from the UH Hilo campus in the Science and Technology Park on Novello Street. Also featuring my supervisor's dog, Zoe, AKA the Sim Lab mascot. In our lab, we do applied research that is focused on insect pests of tropical crops and commodities. Now, many of you are probably already aware of this beetle, either through personal experience or through pictures. This is Acalolepta aesthetica, or as we've been calling it, the Queensland longhorn beetle. It has emerged as quite a beetle of concern here, as it is known to attack cacao, citrus species, breadfruit, kukui, as well as other tree species. The Queensland longhorn beetle, or QLB, belongs to a family of beetles called Cerambycidae, which are the longhorns. Other notable family members that are causing problems on the mainland are the Asian longhorn beetle, which attacks hardwood trees, such as maples, and the citrus longhorn beetle, which of course attacks citrus trees. Now, because they are insects, they are ectothermic, which means they do not produce their own body heat, and their biological functions, such as growth and activity, are dependent on their environmental conditions. And I'll actually talk a bit more about that once we get to our larval rearing for our colony. Their long horns obviously are not horns, but they're actually long antennae, which they use to interact with the physical and chemical world around them. The adult beetles themselves aren't the ones that are actually causing the damage, but it's their larvae, which eat wood. This means two things. One, that they can move around as larvae without being detected, which gives them the potential to be quite invasive. And since they are the ones that are eating wood and can destroy living trees, they can become major problems for an area very, very quickly. So let's visualize the life cycle of these beetles. Here we have an adult that lands on a tree that is attractive to them, either through smell or proximity. They then use powerful mandibles to chew an oviposition pit and lay eggs in it. Now these eggs hatch into larvae, and those larvae eat, grow, and molt, and eventually grow to a big enough size where they stop growing and they pupate. Now, once ready, that pupa will emerge into an adult, find a mate, and then start the cycle over again. Now, the fact that most of this cycle occurs within the tree helps to explain why they're often not detected until damage has been done. It's through that movement of wood that they likely came to us from its native range of Queensland, Australia, which obviously is where it gets its name. Prior to the detection on the Big Island, it was only found in these two specific places, Queensland and New South Wales. Now, among its hosts in Hawaii are the breadfruit, the cacao, various citrus species, and kukui nut. It also infests a host of other trees. Now, the reason we have asterisks by the Norfolk pine and the avocado is because the larvae was present in cut wood, meaning we don't actually know yet if they can infest the tree while it's living, which also means we don't know yet if it's an active threat to those species. However, it is still concerning as cut logs can, and often are, moved around to different parts of the island. This could essentially create a vehicle for the beetles to infest areas that they weren't previously in. So what are the signs to look out for if you believe that you have a beetle infestation on your property? Now, living trees have immune responses in order to protect themselves from pests. One of the potential first signs of infestation includes abnormal oozing of sap coming from that oviposition hole we talked about when we were covering the life cycle. Another sign is girdling, which can lead to the death of the tree. And then finally, you can see wood shavings and exit holes, which shows where the adult beetle has emerged. Now, because of our lab's focus in population genetic analysis, we wanted to use molecular methods to investigate the nature of its invasion on the island so that we can determine how it got here and potentially prevent future invasions. What we do know is that QLB is native to Australia, of course. But what we want to know is if it arrived on the Big Island directly from there, or was there an intermediate source? And wherever it came from, did it come just one time, or did it arrive multiple times? And now that it is here, we need to understand how much it is moving. So once it emerges as an adult, we want to know if it travels long distances away from where it emerged from, or if it mostly just sticks around that area where it emerged, possibly even reinfesting the same tree. Now, what our population genetic analysis has shown us so far is that there is actually no genetic diversity in QLB in the island at the genetic marker that we investigated. This means that there is evidence that the introduction of beetles from outside Hawaii Island has only happened once. This truly could have come down to one female laying eggs in wood that would being transported here, and then once her larvae emerged as adults, they entered our environment. Now, this presented a bit of a problem for us because there was no genetic diversity. We weren't able to use that to investigate dispersal. So we developed a reporting tool so that anyone who found a beetle could report that detection and send a photo in order for us to understand where the beetles physically are. So we received reports from all over the island of beetles, but when you actually look at the photos, the only ones that were QLB were all from this area. Each one of these dots represents a report, and that photo they sent us was used to identify the beetles down to the species level. So as you can imagine, many beetles that were not our species of interest were reported, so having a photo to reference was invaluable. So if we zoom into where all of the QLB were, you see that they are mostly here in Hawaiian Paradise Park, Orchid Land, Ke'au. Though there were 2 reports all the way up there in Hilo, one in Panaeva and one off of Railroad close to the Hilo transfer station and green waste facility. Prior to this reporting tool, we didn't know that the beetle had made its way all the way up to Hilo. So now we know to broaden the scope of where we were previously looking for them. So what that map is really showing us is that the dispersal of this species is actually quite low and likely mostly human mediated. In terms of trapping and lure development, one thing that we have learned in pursuit of developing a trap is that lures are not very effective, at least not the ones that are commercially available for purchase. So as you can imagine, this took quite a bit of trial and error to figure out. However, lures are designed to be species specific, so it's not all that shocking that they might not work for this new pest. There has only been one study on QLB which looked at their attraction to several different commercially available lures. Based on 2 trapping experiments, which tested their attraction to pheromones, one experiment showed that the pheromones were actually a little bit of a deterrent because they caught fewer beetles than the control. And the second experiment showed that all of the treatments were equally bad at attracting beetles. Now, some potential methods for beetle control include systematic insecticides such as soil trenches. Unfortunately, these have not yet been tested in Hawaii and may be quite expensive due to the porousness of the land. Also, as we know, we need to be mindful of protecting our pollinators. Another potential method for control is Bavaria. This is a biological pesticide that uses an insect-infecting fungus, which can kill an adult or larva. This is used on the Big Island to control coffee berry borer, but as of very recently was not labeled for QLB, and thus it would represent an off-labeled use of the product. However, we are now in the process of including it on the label, so this represents a potential future avenue of research. Now here you can actually see the characteristic signs of Bavaria infection in both an adult and larva, specifically the white fungus on the joints in the adult here, and then you can see it on the head capsule of that larva. So because we do still have a lot of work to do, the current best recommendation is to survey, sanitize, and report beetle infestations. The life cycle of this beetle is very long compared to other insects, like for example, the fruit flies that we work on. They live for a few weeks or months in wood, and since the trees exhibit signs of infestation, it is possible to catch a newly infested tree before too much damage has occurred. Once damage is detected, action can be taken and the infested wood can be removed and then chipped in order to prevent further movement of the beetle. So our lab is currently pursuing other QLB projects, one of which is determining beetle infestation from larvae and frass. Since there are many species of longhorn on the island, it is super difficult to tell their species based on larval morphology. At their early immature stages, they look incredibly similar. But we're developing methods to do genetic analysis on the larva so that we don't have to wait for it to emerge as an adult to tell what species it is. Knowing this would allow farmers to know what actions would be appropriate to take. Additionally, we want to be able to genotype frass to determine if it is frass from QLB versus a different species. We're also in the process of establishing an experimental research colony that we can use for the lure development and also testing different insecticides. This will hopefully enable us to provide recommendations for specific pesticides that are effective at controlling adult beetles and protecting trees. Establishing a QLB colony is significantly more complicated than one would expect. We start the process by sexing and then pairing to adults, allowing them enough time in our breeding chambers for the female to oviposit into the kukui, wood bolts that we have provided. We then keep those bolts in bins until evidence of larvae is apparent, i.e. frass or girdling. We then very, very carefully extract the larva from the bolt and put it in its own artificial larval diet jar. It feeds on the diet as it grows, and once the larva is in what we believe to be its final instar, what we have been doing has been chilling the entire jar down to 10 degrees Celsius. The idea behind that is that this would encourage the larva to pupate, and the pupae is then transferred into a tube with a wet paper towel to keep it damp. However, we are also testing to see if they will pupate at room temperature. The pupa is then carefully observed until it emerges as an adult, and then the process begins again. Because so much about this species is unknown, our process evolves and changes constantly in order to optimize survival. And we are continuously learning. Here you can see one of the bolts that has been oviposited into, as well as another technician who is on the project, Amberly, observing an adult QLB right on the end of that stick. Here you can see two adults beginning to engage in mating behavior. Our current colony houses approximately 30 larvae and 20 adults. Now, one of the issues that we ran into early on in our colony building process was being able to determine the male versus the females of this species. Typically, the male have longer antennae. However, that can be very relative to the size of the beetle itself, and sometimes we receive beetles with missing or partial antennae. One of the first pairings that we had ended quite tragically when we learned that if you mistakenly pair two females together, they will attempt to kill each other. So we needed an additional way to sex these beetles. Oh no. Okay, sorry guys, I have to share my screen again. We're having some monitor technical difficulties. Give me 1 second. All right, I'm hoping that you guys can see that again and we are good to go. Let's see. I'm assuming if no one's gonna say anything that we're good. Okay, so like I said, it ended pretty tragically when they did attempt to kill one another. So we did need an additional way to sex the beetles. So we stuck them under a dissecting microscope in order to more closely study their abdominal structures. Here you can see the male on the left and the female on the right. You can see the male has more of that V-shaped tip with an excess of bristle, where the female has more of a rounded shape with a more prominent nub at the end, which hints at the existence of her ovipositor. Now, having our colony has also allowed us to gather data on how many instars these larvae go through before they pupate. With the help of two other technicians, Amberly Buer and Stephanie Gale, the width of the larval head capsules were measured over time. I then took that data and visualized it. And as you can see, we've got a pretty distinct pattern going on. You can see the frequency at which the different widths were encountered. And though we are still actively performing the statistical analysis on this data, this has already provided us huge insight on the distribution of larval instars. And of course, with everything going on in 2020, we've had to collectively and creatively modify our rearing methodology in order to keep each other safe and to minimize in-person time in shared lab spaces. So if you want to help us, if you have a live adult, you can bring it to us so that we can use it to either supplement our colony, or if it's dead, we can still use it for genetic analysis. We would require some data, such as when and where it was found and what it was found on. Now, if you point your phone camera at this QR code, it'll take you to this website where you can complete the report form on your phone, and also so you can submit a photo of your beetle capture. I will have the QR code again on my final slide for anyone who would like to use it. Once again, you can bring the beetle here and we can receive and process it. Now, if you can have a live beetle, we do prefer that you bring it in a glass or hard plastic jar, as those powerful mandibles can be used to chew through cardboard and soft plastic like Ziploc bags. And we really would prefer it not getting away into our lab space. Also, with regulations in place during COVID, we may not physically be at the lab every day. So if you do have a beetle you would like to bring us, please email me ahead of time so that I can ensure that it will be processed in a timely manner. With that, I'd like to thank all of you for listening, and I would like to acknowledge all of our collaborators. As you can see, there's an incredible amount of work that is devoted to protecting Hawaii from these invasive pests. None of this work could have been done without the hard work of so many of my coworkers at the USDA and our collaborators at BISC, HDOA, and USGS. I would also like to thank the East Hawaii Cacao Association and Hawaii Chocolate and Cacao, who have growers who have very graciously allowed us to attempt to trap beetles at their property. I would like to thank the organizations who have funded this work, of course, the Hawaii Invasive Species Council and the USDA, and of course, Dr. Shenna Sim and Lisa Canali and the rest of the TCBES faculty for providing me with the foundation to do meaningful work here in Hawaii. Once again, there's that QR code and my email if you would like to contact me about any beetles, and I would love to answer some questions.
For a great presentation. Keena, please feel free just to unmute your mic and ask your question.
I have a question.
Yes.
Okay. Keena, great presentation. That was very interesting to hear about. Can you please tell me more about the molecular part of your project? Is there a certain part of the genome that you study or plan to study?
So what we've primarily done so far is based on CO1 sequencing of these beetles, just establishing if the adults are what we thought that they were. Like I said, I think we're going to definitely start focusing in on that frass kind of direction of things, only because it would be so incredibly helpful for a farmer to call us, just say, hey, can you come out to my farm? There's something infesting my tree. I don't know what it is. And for us to just be able to collect the frass and then answer that question for them, as opposed to potentially doing something destructive to the tree, like having to take a limb only to find out it wasn't this and it would have been fine.
Thank you.
Thank you for your talk. It was really good. It's kind of fun to see what you've been working on. I was really curious, what's the success rate when you do get a couple to mate, and then how many eggs and how far does that go along? How big's your population now in the colony?
Yeah, so we've been quite successful getting them to oviposit into the wood that we do provide them. And so we do have, I think, 29 larvae on artificial diet right now. What we are struggling with the most is actually getting them to pupate and being able to complete that life cycle. I think we have had success a very small handful of times in that, only because they seem to really like to pupate in the wood, which makes sense. That's what they do. And so we're trying to find that perfect balance to get them to pupate in the artificial diet so that we have the capacity to rear more of them.
Keena, did you develop the QR code and the form and stuff for this?
No.
How is that done?
So no, that was actually Shenna did. She's worked very, very hard on that form and being able to do that mapping so efficiently. So I'm not sure if she would like to answer that question.
Yeah, actually, I got a lot of help from someone who works at Colorado State. His name is Jason Zerish, and he's the one who actually helped me set up the form. It's a GIS online form. But as far as a QR code, all you do is go to like qrcodegenerator.com, and then you put in a URL, and then they'll spit out a QR code that then directs you to the URL that you provide.
Natural predators. Has there been any known, is there any known natural predator in Australia? And then has there been any sightings of the beetle being eaten by a bird or, a different kind of predator that they might have here in Hawaii?
So unfortunately, these are very, very poorly studied beetles. They just don't present as a pest in Australia. So there has never been a need for them to study biological control or who the natural predators are or anything like that. And I don't believe we have seen any sort of predation of them here naturally at all. So, and that's part of the double-edged sword of working on something that is so unknown and so new to the environment. It's great because everything we learn is important for our general knowledge of the beetle. However, we have very little foundational work to, back or to give us the right direction of things. So it really is just uncharted territory.
Kind of a related question. Based on the habitats that live in Australia, do you think they're going to be able to expand into other areas of the Big Island or across the state? Looks like they started in Puna, and they're an invasion that's underway. And what are going to be their limits?
Yeah, so that's a great question. When we were more actively trapping, we did deploy traps up the Hamakua coast as well. And we never found any evidence of beetles up there. Similarly, they seem to kind of end after Curtistown area. They're not really going, or we have not gotten any reports any higher in elevation than that. So yeah, we really are very curious as to how this beetle is going to disperse in the future. And it does seem to be heavily human mediated. Like I mentioned, one of the beetles in Hilo was quite close to the green waste transfer station. So it's possible that somebody may have been moving infested wood, and that was the cause of that beetle being there. It's a very good thing for us that they appear to have very low dispersal. Hopefully that means that we can more effectively handle this before it gets even more out of control than it is.
I have a question. So, what would you say you've learned from pursuing this work, and how would you apply what you've learned to your position in the future?
Sure. Oh gosh, I have learned so much from these beetles. It has been wild. I've only ever worked on either Tephritid or Drosophila fruit flies, so this was a completely new area for me, and it presented a very different set of challenges to overcome. And I think something that has been one of the most important things I've learned is just going through that like very basic scientific process of trial and error when you just truly don't know what's going to work, such as when we were first formulating the larval diet and things like that. And kind of just being able to take a project, move forward, and critique it slowly so that you can both comprehensively understand what is and isn't working, as well as be able to kind of keep yourself from making those same or similar mistakes in the future. And I absolutely think just having that fundamental kind of way of thinking can be applied to all of the projects that I work on in the USDA. Like we do very much applied research. So it's great to be able to kind of tackle these issues, look at them and figure out how they are going to be applied in a grand scale and help our collaborators, help our community, things like that.
I have one more question. So this is more like hypothetical. Let's say there's a brand new invasive species on the island. We know nothing about it. What's the first thing you would do? And how would you have done the response to QLB a little bit differently?
Oh gosh. Oh, that's a great question. I think something that I would have done, I think a little bit earlier, I would have liked to have started the establishment of a colony, only because now we are understanding how important that is for our next steps. So I think, yeah, going forward, I would, if it were a brand new species, I would put an emphasis on catching live or trying to collect live samples, be it through community bringing it into us. Probably not trapping because we're figuring out that's a lot more complex than we necessarily thought in the beginning. And just, yeah, having a big source of live samples to be able to source from, both for the genetic component as well as the, ecological component.
Thank you.
Hi, Keena. I have a question. How much is known about its host range, host breadth within its native range in Australia?
Nothing. It's truly nothing, which is wild.
Yeah, it seems like if it's been found in the north, in Queensland, as well as New South Wales in the south, there's a good chance that it's attacking a variety of maybe quite different trees there as well.
Yeah, absolutely. And it's definitely something we would like to pursue in the future, both getting live samples and getting a little bit more information from collaborators in Australia. Obviously, that's a little bit more challenging now in COVID times than when we were first talking about it, but it's definitely on the plate for us.
And just a follow-up question, is it considered a pest anyplace else in the Pacific?
No, we are the only place that it is considered a pest so far that we know of.
Okay, thanks.
I think we've gone through the questions, so thank you very much, Keena. I will copy the chat and send it to you so you can have it as a keepsake. And if everybody, if you don't mind, on the count of three, just unmuting yourself and saying thank you, I'd Appreciate that. So 1, 2, 3. Thank you.
Thank you.
Congratulations.
Thank you.
Thank you everyone for coming. I really appreciate all of you. Thank you.